thank you. Thank you for staying at this point and letting me tell you a little bit about LifeEdit Therapeutics. So we are a gene editing company. We're based in RTP in North Carolina. And over the last few years, we've built a platform approach to gene editors. So this is a platform that includes nucleases and proprietary base editors. We started building this back in 2017. I'll show you a little bit about the history of the company. And we've really got to the point now where these are ready to be moved forward and start to think about them in the context of gene editing therapeutics. So we've started to do that. And at the same time, because it's a platform, because we have many nucleases within this, we're also looking at opportunities to democratize the access to these enzymes. So where did we come from? We actually came from an ag company based in RTP. Company called AgBiome, who had built a microbial collection for use with agricultural applications. They started this collection back in 2014. They made the decision at that point to do whole genome sequencing on every strain, and that resulted in a really mineable database. And so as CRISPR started to develop, they recognized that it was a good time to start looking in the database and see what we have. And so the group that's now LifeEdit Therapeutics did that. They identified thousands of CRISPR systems. And at the end of last year, we spun out of, or in October of last year, we spun out of AgBiome and became an Elevate Bio portfolio company. With that transition, we took with us all the IP that we generated at AgBiome, and we continue to have access to the collection to mine it for systems, gene editing systems that can be used for therapeutics. And so this allows us to continue to build this platform and look for all kinds of gene editing systems we might need in the future. So we've become part of Elevate Bio. And where do we sit? So Elevate Bio is an accelerator of cell and gene therapies. They've got a number of technologies, from gene editing, which LifeEdit brings into the family, all the way through iPSCs, protein engineering, and full manufacturing at their base camp facility. Their intention is to take all of these technologies and platforms and use them to accelerate the cell and gene therapies of partners, to build their own therapeutics internally, and to build a manufacturing capability. How do they do this? Really, Elevate Bio takes a number of different approaches, but really the driver is integrating all these different technologies in a way that will accelerate and disrupt the cell and gene therapy space. LifeEdit's super excited to be a part of the Elevate Bio family because what it brought to us was the cell and gene therapy development experience and expertise that we didn't have. So we really built a center of excellence for gene editing. Elevate Bio brings us drug development. They bring us manufacturing, in particular for AAV. They bring capital commercial experience. And so between where we came from with AgBiome, the platform we've built in the middle, and all the support that Elevate provides us, we're now in a position where we can start moving forward with the types of work that we want to do. And so what is this allowing us to do? It's allowing us to do all the things that we had set out to do. So we continue to build the platform. As we've built this platform, we've tried to identify smaller sized nucleases, which would allow us to deliver them by AAV. We can then leverage the AAV capabilities at Elevate Bio to be able to do that. We're moving them forward to validate our systems both preclinically and ultimately clinically. But by partnering with others, we're also able to look at these systems in the hands of others, in other people's pipelines. So we have a fantastic leadership team. Our CEO is Mitch Feiner. He's also the CSO at Elevate. We're joined by Kareem Reeder, who's our Chief Operating Officer and their Chief Business Officer. Ted Elich and myself came from AgBiome. We had joined AgBiome specifically to spin life edit out, and we were recently joined by Catherine Woodburn, who brings all her preclinical and delivery experience to the organization. We started last October. There were 5 of us. We're at 30 people now. We're growing. We're close to 100 by the end of next year. So lots of exciting things going on. So we've put the company together. We've got the leadership team. We're building the scientific teams that we need. What are we doing with that? So we're starting to think about, how do we use this collection of nucleases and the approaches we can take to try to address some of the limitations that are really there for CRISPR. And the way we're trying to do that is by mining the natural diversity or leveraging the natural diversity we have access to. So, as I mentioned at the beginning, we identified thousands of systems. We then culled that down really to single protein effector systems targeting DNA, and we've got predominantly type 2 and type 5 nucleases. As we identified these, we noticed that we had massive variety and diversity in terms of the PAM sequences, which then allows us to access a much broader array of genomic sequences for editing. All of the microbes we mine are non-pathogenic, which we hope will result in reduced immunogenicity risk. And at this point, we've got about 60 of these RNA-guided nucleases identified and characterized. So just to give you an idea, you think about this, well, okay, we've got thousands of these systems. We've got 60 of them with PAM, different PAMs, but they're all type 2s. How diverse really is this collection? Well, very, as it turns out. As you look through the kind of phylogeny, they're quite diverse. And then if you start to do pairwise amino acid comparisons, you see that these are not, as you move from one enzyme to another, they're quite different. And we see that then in the activity. Not every type 2 RNA-guided nuclease is the same. And in some cases, we've got as low as 1% identity. We've been able to take a number of the lead enzymes and start to look at them in mammalian cell lines. And so we have data in HEC293 cells. We've done a lot of work now in primary T cells, and we've also been looking at them in mouse fibroblasts as we take them into various in vivo models. And what we see is really quite nice editing across the board. This is a sample of the data. We actually look pretty broadly at a number of genes early on, kind of a high-throughput screen, and pull out the most robust nucleases to then be able to take them through to the more detailed screening. We've looked at these with different delivery methods, both mRNA and RNP, and we're just seeing excellent editing. We've also been able to show that we can do multiplexed editing. This is an example of a nuclease LEG14. We've looked at it with TRAC and B2M in primary T cells. So all our editing data is NGS, so we get we do a next-gen sequencing, but we've also started looking at the functional data, and you can see here that's consistent with the NGS data that we're generating. So having identified the nucleases and understanding that we had quite a lot of PAM diversity, we started to think about what are the other editing modalities we would want to be able to do. So we've done some HDR knock-in work, and we're doing more of that now to really understand how large an insertion we can do and how robustly we can do that. And we've done some lovely work in CRISPR-A and CRISPR-I. But I think what's been really exciting for us is our ability to develop some C and A base editing technology. So the C base editors, we mined for the deaminase out of our collection. That was something we could pull out of the collection and use with the nickase. We've resolved the issues of activity. These are very modular, so we can take the nuclease, the deaminase, and kind of work based on the edit we want to make. We see no double-stranded breaks and low indel formation. On the A base editors, we obviously use the pioneering work of David Liu to kind of fast track us down that path. In this case, you can't find these. These are not naturally occurring. And so we've been doing the protein engineering to really bring those forward. So both of these base editor platforms continue to evolve. But we're getting some very, very nice data. So what are our next steps? We're clearly in the very early stages. We want to continue to identify and characterize and optimize our new nucleases and base editors. Obviously, we want to start taking these forward in vivo, and we should have that data early next year. And we continue to think about what are the next gene editing platforms that we want to be able to develop. And that will be driven very much by what do we want to do therapeutically and what are the edits we're going to want to make. We're building our therapeutic pipeline. The in vivo data we should get early next year should confirm some of the directions we're going to go with that, and we hope by the end of next year to have development candidates selected. And finally, we're in the midst of really discussing with partners how we can use our gene editors as enabling technologies in the cell therapy space, as well as in vivo opportunities. I think that's it. Any questions? That's easy. Thank you.